All right. And you can hear me all right, Dev? I'm just going to use you because you were speaking.
Yep,
Awesome.
loud and clear. Thank you.
And my slide, I just advanced my slides and my screen is doing all it's supposed to, correct? Awesome, perfect. I always hate when I get like 3 slides in and someone's like, hey, Sean, you forgot to resume share. So anywho, today we're focusing on the Genomics Workbench, specifically our long read tool set. Devin kind of said case study. I think this is a little more training and or so, at least my focus a little bit, but we do have a small subset of data that we will go through and whatnot. You could just imagine some of these larger data sets would just be a challenge to do in our hour and a half. Of course, it's important to have our legal disclaimer that these tools are intended for molecular biology applications and not intended for diagnosis, prevention, treatment of disease. Pardon me. Additionally, I just also like to point out, I think Zoom still looks like this on your screen. However, you should see on the bottom of this screen a chat, a raise hand, and a Q&A. And just today, for today's purpose, we'll potentially be using the raise hand and the Q&A. If you have a question, please use the Q&A. It just makes it a lot easier to track, more so than the chat. So if you wouldn't mind, again, if you have questions, please use the Q&A on the bottom of your screen during today's presentation. All right, so want to give everyone a quick background of what we're going to cover today, and then also just a quick agenda of all the different steps that we're going to be doing. So really, from a background point of view, the long read is actually, our long read support is actually from a plugin. It's a collection of different tools to be, really focusing on using like Oxford Nanopore or PacBio data. Then those reads tend to be a bit more error prone. And so what we've done is we've really, in a way of wrapped, or I wouldn't say wrapped is a bad terminology. We've taken some open source functionalities or algorithms and sort of compiled them in a way that works within our application. Those are like Mini-Map2, Mini-ASM, and Raccoon. And so today what we're going to be covering is a quick overview of the Genomics Workbench. Once we talk about the Genomics Workbench, there are some plugins that we're going to be using. And because of that, I just want to talk briefly about how plugins are installed. And then really it's an importing our data that's going to be required for today's analysis. And that's really going to be a set of ion torrent data, an Illumina data set, and a reference genome. Once we have that, we're going to be looking at doing like a de novo assembly of this microbial sized genome using the MinION data. And then we'll go back through and we'll look at how we can improve that assembly from the long reads by polishing with short reads, short high quality reads. And then lastly, I can talk about how we can map reads to a reference and other visualization in visualizing an assembly. There's some use cases in this to be able to do some of that type of functionality as well. Some of it is a bit time dependent, so we'll see how far, if I can get into a couple other things as well. I always like to give everyone a brief overview of sort of the Genomics Workbench in a sense of like what tools, what functionalities can it sort of support. And actually, I might use this time if people can use, I'm going to actually say the chat. So unlike I asked for before, maybe for Deb to be able to capture, if we look at this set of tools, we're going to be trying to do different trainings in the upcoming months. Next month, it wasn't on the list, but I'm going to focus on doing some ATAC-seq type of single cell analysis. And so if we look at this list of functionalities, if there's anything that really seems interesting to you guys, if you want to jot that down in the chat or in the chat, that would be wonderful. So, I mean, if we talk about different research areas, I'll start from the upper right-hand corner, like the 1 o'clock, if we had a traditional analog clock. This is where, we have a bunch of different resequencing types of functionalities, trimming, mapping reads to a reference, variant callers, things along those lines. So your SNP, indel, structural variant type of calling functionalities are actually part of this Genomics Workbench as well. Continuing around, there's actually a lot of general bioinformatics types of stuff, traditional as I like to call it, right? Alignments, phylogenetic trees, Sanger assembly, primer design, blast searching. So again, a host of other types of functionalities. We do have a premium version of our workbench that comes with a couple additional functionalities, one of which being microbial genomics, types of plugin and functionality where you can do things like amplicon-based metagenomics, 16S, 18S types of analysis, whole shotgun metagenomics, functional metagenomics, or like typing in epidemiology. And then kind of continuing along epigenomics, so ChIP-seq, bisulfite sequencing, a whole host of expression analysis functionalities from bulk RNA-seq, microRNA analysis, single cell analysis, and then rounding out the top here, the de novo assembly. Again, what we're going to be covering today are a couple of different plugins that are used, and I didn't outline them in sort of the core functionalities that we see in the workbench here. So if there's anything that you guys see really interesting or whatnot that you guys would like covered, it'd be nice to get that type of information as well. Some reasons why to sort of use the Genomics Workbench, if you guys aren't or haven't used the Genomics Workbench in the past, it's wizard driven. And so what I'm going to be showing you today is, not only can we handle these long reads, but hopefully it appears to be, easy to do the analysis. Again, everything's, wizard driven, where you'll be asked for, an input, how to handle results, where are we saving the data? And then if anything is configurable or parameters that we can, change if we had a workflow, then we would be able to do that within this workflow manager as well. Additionally, when we talk about science, I think it's important to have outputs that are graphical sense, just because, this is what we tend to put in publications. I believe they say a picture's worth 1,000 words. And so we can really convey a lot of what's going on in our experiment when we have the ability to export things like heat map, bar charts for our metagenomics, phylogenetic trees with metadata annotated on top of it. And you can see a slew of different like RNA-seq types of functionalities like PCA plots, Venn diagram, and then this is actually a PCOA plot from metagenomics. So really, I think when we talk about the, having an analysis tool, the fact that it's wizard driven and it can produce really good graphics for my publications are really what makes the workbench something that's quite useful for everyone to use. And then the last thing I kind of want to just point out before I jump into like an overview of the application is that we're really just talking about a standard single workbench today. But if anything, if you interview one at a larger organization and wants to think about scale, we do have ways of scaling either through high performance computing environments or using AWS cloud types of deployments. So that was enough of my cool sort of background. I do like to just kind of give everyone a sense of where some of the key areas are to know, because I'm going to be jumping, into the application so we can actually go through this in the Genomics Workbench. And so I always like to point out the import tool in the upper left-hand corner. This is where we'll be importing reads, reference sequences, and things along those lines. Just below that import function, you'll notice the navigation area. It's a flat file system. It's just where we're going to store data. So everything that we generate results from will be stored in this location. And then we can see just below that is our toolbox. And this is where all the tools are that we have access to. There's a couple other ways people will launch tools as well. But this little quick launch up top can be really useful if you're trying to find a tool and you're not sure where it belongs in the toolbox. And I'll show that as we get into the application. And then the last area that I wanted to point out is just this upper right-hand corner. This is where the plugin manager is, where you'd be utilizing to install different plugins into the application. A reference manager for downloading model organisms, a download function for downloading sequences at like NCBI, SRA databases. And then we also see over here on the far right, the ability to sort of manage workflows, manage and or build workflows. So that's my quick overview of the Genomics Workbench. I just thought it would be kind of important just to give everyone a quick lay of the land and just an understanding of the other research areas that we can kind of facilitate within the workbench. Again, I mentioned that these are plugins, and so what we'll talk about is how do we install plugins into the Genomics Workbench to add that long read plugin and our whole genome alignment functionality. I'll show this in the workbench as well real quick, just so you can see it live. But again, I talked about in my slide previous that there's a plugin manager in the upper right-hand corner. When we click on that plugin manager, it opens up a dialog box where we can either manage plugins that we have installed, or we can download plugins that we already have installed. Or sorry, or we can download plugins that we haven't installed. So for instance, if you can see, this long read support was under my manage plugins, and that's why there's an uninstall or disabled. And then the whole genome alignment tool, we can see has a download and install button that's active. And so this is under the download plugins type of functionality. And all we do is click on that download and install. That will install the functionality into the workbench. And then when we close the dialog box, it will ask to restart the workbench. When we talk about the long read support, this is still in beta. I expect it to be coming out shortly. The functionalities that we can see within this long read support are going to be like correct long reads, de novo assembly of long reads, mapping long reads to a reference genome, polishing with reads, pardon me, and then RNA-seq for long reads. So the correct reads is obviously an error correction type of tool. One caveat to that, like we actually build that error correction of long reads into our de novo assembler, so oftentimes not required to do before doing the de novo assembly in our tool. But it does give you the ability to do error corrections and maybe correct data to go out and export into other tools potentially. The map reads to a reference, obviously, if we have a reference genome that we were looking to map our reads against, this would be the tool that we want to use. And what we will be using today is the polish with short reads, if you will, where we have a set of Illumina data, and we're going to use that after we've done the de novo assembly to really polish and refine the assembly that we generated. And then last, if you do have like Oxford Nanopore or PacBio RNA-seq data, there's an algorithm for quantifying transcripts and genes and transcripts for long reads specifically. And again, if we were to just see this in the workbench, pretty simple, where we click on the upper right-hand corner. Again, it gives us this dialog box where we can interact with the plugins that we've already downloaded. And again, because of for today's talk, I have all of these already downloaded, but we can also see other additional plugins that are available. If we look at like annotate with GFF, there's no additional like sort of information here in red. This is a freely available plugin. Something like CLC MLST module, you'll notice that this is a commercial plugin that requires an additional license. But if we install any of these plugins, so we click that download and install, you do need to accept the end user agreement. And once you've finished the installation, you'll notice it'll ask to restart the workbench, and no functionalities will become available until you do restart that workbench. So augmenting the application, adding different plugins should be really easy to do. It's all done right from within the application and just managed through the plugin manager. Now, I always like to have an output. So as I mentioned today, our goal is really to do some long read assembly and using some short reads to help polish up the assembly. And I always like to give you sort of a sense of, well, and the screenshot didn't come out as good as I was hoping, but what we can see is in this top read map or assembly, we can see all of these different tick marks in the red and the green area. These are representing errors within the genome or mismatches in the read. So this is actually an example of that assembly without doing any error correction. And we can see that once we've done some error correction, how the assembly has been cleaned up, where it would actually be possible to call potential maybe mutations, where we can see there's a difference in this location, a difference in this pileup of red reads. But again, what we can see very clearly is that, the assembly or the down here has a lot less errors in it. And that's because we are able to use the error correction and the polishing with the short reads. It's always nice, as I said, to have an understanding of what our goals are before I sort of embark on our journey. The data for today's talk is just from a study that examined the feasibility of using Oxford Nanopore to fully assemble and resolve bacterial genomes, including plasmids. So it contains long min-ion reads and Illumina short read sequencing data from 8 bacteria isolates of 6 different species. Today, we're really just going to focus on one of those just to make it a bit easier. And this is the species that we're looking at. And the strain has 1 chromosome and 5 plasmids. That contains, the sequencing data contains about 7,000 MinION reads with an average read length of 12,660 bases. And then we're also, like I said, importing a set of Illumina data that have been downsampled from 3 million to 300,000, again, just to kind of reduce runtime. And again, this is also available through a tutorial, so that way you can go through the same types of steps as well. And then lastly, we're going to bring in a reference genome from this specific bio project that we can use as a reference to do some additional types of functionality like a whole genome aligner and things like that. So I always like to start out with bringing our data into the application. And so, as I mentioned, our first thing that we're going to have to do is import the reads into the application. And so what we're going to be doing is using the upper left-hand corner where it says import, and then we'll break it down into several different options like standard import, tracks, your NGS sequencing vendors. Then you'll see like RNA spike and a whole host of other types of data that we can bring into the application. Today, we'll be focusing on Illumina, Oxford Nanopore, and the standard import. And the data that we're going to be bringing in is going to just be we're going to be importing the Oxford Nanopore data by selecting the file import Oxford Nanopore and adding the files from our FASTQ file. Once we're done with that, we're going to open the same import function, except select Illumina and import the paired reads for the Illumina data set, which contains an R1 and an R2, one for each of the sets of paired reads, one for the set of a forward and reverse for the paired reads. And then, as I mentioned, lastly, we're just going to use that standard import to automatically import the reference genome. And our sort of navigation area will look like it does down below. So as I said, importing and everything in the application is very wizard driven. So we'll just select our Oxford Nanopore as our first set of files that we'll import. And I'm just going to say add files. And you can see it just gives me all of my all of my file structure. And I will just set this update. And so here we can see our MinION reads, as I had mentioned. We'll just simply say open. And now that it's pointing to the file, we can simply click next. We're going to save the data, and I'll just leave it at the root directory. And so I'll simply click next. And as I said, we'll make a folder called long read. And hit finish. So importing data is pretty straightforward, and I'll just do the same for the Illumina data. This time when we click add files, I'll be selecting the R1 and R2 for the downsampled Illumina data set. Important when we import Illumina paired data that we want to look at these general options and just verify that the paired reads is checked. And what this will do is excuse me, it'll interleave the R1 and R2 file into one object, which just makes it a lot easier when we start dealing with the files during the analysis. And now that I've created the folder, I'm just going to simply select the long read tutorial folder and select finish. And now that imports the Illumina data. And as I mentioned, the last one we really need to bring in is that reference genome. And again, we'll be clicking on the import, but now using the standard import. And I'm selecting this FAST file. And once again, I will simply select the long read folder that we are storing the data in. So what we can see is that under the processes that I've imported my standard FAST file, my Illumina data, and my Oxford Nanopore data. And we can now look at that long read tutorial folder and see it looks exactly how it did in that slide where we have our MinION reads, our downsample Illumina, and our genome that we'll be using as a reference. If we want, we could of course double click on any of these objects and inspect the reads. And what you'll notice here is like the read name, the AC, T's and G's, and the quality score at that position for the base. Additionally, as I mentioned, the Illumina data gets merged into one file, and we can see that with the name of the file looks like R1. However, it has parentheses in its name. Has paired in parentheses in its name. And we can see when we look at the name of these reads that it's the R1 and then the R2 for the same mate. And so what it does, if you will, it zippers those files together. And so you have R1 and R2. If there's any failed, reads, we would end up with like an orphaned file that contains singletons. And not much to really say, but we did import that reference genome. And as I mentioned, this is going to be 6 different sequences, one being the bacterial genome, and then the other 5 being plasmid sequences, excuse me. So that gets us to the point where we've imported our data for the to do the required analysis. And as we start to progress, we're going to look at how we can do the de novo assembly for the genome using the long reads. And at first, we're simply going to only be using the long reads. So let's go back into the application. So if we look at the tool, after we install the long read support tool, we have a set of tools in our toolbox called long read support. And in this, we have a set of tools as I showed in my slides. But right now, we're going to be looking at the de novo assembly of long reads, and we can simply double click on that algorithm. When we do so, the navigation area is just simply opened up, and now we can select any of the reads that we want to use in the assembly. Additionally, if you're unsure of like, whoa, what am I supposed to be selecting here, or what step in the workflow or in the algorithm am I in, we can always look in the upper left-hand corner here, and you can see it says select sequencing reads just to help clarify the step that we're at. And in this case, I have one data set that we're looking at, and so I'm going to just push it over to the right and click next. Now, when we get to the de novo assembly option, you'll notice that there's an option for polishing reads. By default, that's going to be toggled on. And so we will be using the long reads to polish themselves, right, in a way where we can actually help clean up the genome by just using the reads themselves. And what we're setting out here for the output filter is that the minimum contact length is set to 1. And I mean, again, we're going to just click next and be able to create a report, create an assembly graph, and then simply save those results. I'll actually generate a new folder by selecting the long read result tutorial root directory and now clicking on new folder up top and just call this results. Whatever we want, but I'll call it results with mid-ion only. And we can see that once that starts to process, we can go to the processes tab and we can see how far along this process is. Additionally, from that little arrow, we can stop or show the log information. So here you can see that it's going to give us a time lapsed in the log. And this is going to say when we started the algorithm and the information about what step it is in that process as well, like preparing the reads, running the CLC overlap assembler. And again, you can see this is going to be based on Raven. And just to give you guys a sense, I mean, again, we're looking at a bacteria genome, so it's not overly huge. But again, I am using a laptop from 2017 with 16 gigs of RAM to be doing this analysis. And I don't believe it takes much longer than just a couple minutes. At this point in time, if anyone has any questions or whatnot, it would be great to stick them in the Q&A. Gives me a quick time to give a glance at the Q&A if there's any questions that have come up.
Sean, one of the things that you touched upon, but maybe we can reemphasize in terms of introducing new features in CLC, how often does that happen?
Oh, well, the Workbench has at least one major release a year, sometimes 2, and also several point releases. When we talk about the plugins, and that's sort of the nice thing is that we have that plugin manager, so we can actually release anything that comes along, mid release cycle as well. So that's a nice function and feature, but there's several updates yearly, and of course that's even for plugins as well as the core functionality.
Awesome, awesome. And again, as Sean was pointing out to our audience, the Q&A box is at the bottom of your screen, so feel free to type in your question. We'll take even more as we move forward. Back to you, Sean.
Thank you, Deb. And so we can look at the log file. Again, it really didn't take much time, a minute and 35 seconds to assemble that 7,000 reads on my laptop. And now, just to always take a quick look at those results. Personally, I like to look at the assembly report to give myself a good understanding of, how well did the assembly go? What's the sort of quality of the data? And if we look at sort of the contig measurements here, we can see that we created 6 contigs, which would make sense, right? There should be 1 bacteria genome and 5 plasmids. So that's always nice when we kind of have the number of contigs that's expected. We can see the minimum length is about 3,100 bases, and the maximum length here is about 5 million bases. This is really nice as a sense to have a report of how your assembly went. You get nice little graphs as well. So we have a bar chart for the number of contigs that fall within certain breakout ranges. Additionally, the number of contigs that are created and their contig length sum as well. Anytime you have something like this in the workbench that you want to export, you can simply click on the export function. And in this case, it would be a report. So we would probably export this in something like a PDF or JSON file format, and we can simply export this to our desktop, for instance, and it'll look exactly how it does on screen. But again, anything that you want to export in the application as it pertains to a file would be exported through the export function right here. Additionally, it did generate a contig list. So all of those sequences are also generated as well. And we can see here that at least 3 of these are shown up as being assembled in a circular format. And these top 3 are shown to be in a linear format. And the reason why I can tell that very quickly is that the bottom 3, I don't know if it's difficult to see on your screen, but have 2 little carets at the beginning of the sequence, and those would also exist at the end, showing that there's a wrapping of that genome around the origin. So that's just a representation of what would a circular sequence. And so at least from the assembly point of view, 3 of the plasmid got assembled correctly in a sort of a circular point of view. 2 of them didn't. And then we have our sort of bacteria genome that we assembled. And then lastly, I kind of look at this as a table format. So there's several icons down here in the lower left-hand corner. One being a table, which just allows me to look at those sequences, the length of the sequence, and then also, what type of format is it in, linear or circular. So hopefully the de novo assembly seems pretty straightforward. Again, I mean, we didn't really have to do much more than double click on the de novo assembly for long reads in the lower left-hand corner, select our reads that we're assembling. And again, I had 1 set of reads that we were assembling in this case. And so then once we clicked on that, we were able to, leave all the parameters pretty much as default, at least for this data set. And then, able to generate our 6 contigs and a nice report that we were able to use for sharing information with colleagues, putting in our papers, etc. So once we've downloaded those contigs, one of the other tools that or plugins that I had mentioned wanting to install or installing was a whole genome aligner. This is a MAUV-like algorithm brought into the application and really just gives us the ability to do alignments of large genomes. And so it's not a bad idea to do the whole genome alignment of these 6 contigs to our reference genome that we have. And so what we can simply do is create the whole genome alignment. And what we're going to select are the results that we just created. And so the results from my de novo min-ion only. And again, I'm going to leave everything as default for now, but what we can see are like, the minimum seed length, if we allow mismatching within the seeds, minimum alignment block length, and then rearranging the contigs. And the reason why, I'll leave them as default because it actually works pretty well for this data set and many others. But again, it's important to understand that, even though it's a GUI application, you as the scientist are able to make changes to how this algorithm behaves. And additionally, if you're trying to understand how something works, you can simply hover over. And if it gives you enough information, that's great. If not, you can always click on this help button in the wizard. And here you can see it brings us directly to the help manual for the whole genome aligner. And if we wanted to understand anything more about these specific settings, we can look at the information within the documentation. So again, you can always click on that help button, but what I'm going to be doing is just using the default settings for today. And now I would just simply select a reference genome. You can see it already has one selected just because I've worked on this before. And I'll simply save the output here. And click finish. So again, this is now going to calculate initial mismatches and create us an alignment of our of all 6 contigs with the reference genome. Again, all of this being run on my laptop, you can see it still happens pretty quick. And when we double click on the whole genome alignment, we can see that we have our different contigs here. Look, this is UTG 2140, and we can see this is our long read contig 2138. And so we can easily hover over these and see the different contigs that we created along with the reference sequence that it was matching to up above. Now, it's nice to have this type of output, and of course you can zoom in, right, where you can actually see the ACTs and Gs of the alignment between the genome that we brought in and our reference sequence down below. Or sorry, our contig sequences down below and our reference genome up top. Now, honestly, to look at these whole genomes, it's nearly impossible to go through and manually inspect these. So I generally would use create an average nucleotide identity comparison. This is going to allow us to see sort of the how similar are our sequence, if you will, and really be able to give us an idea of because we know that this is a reference to quality of our assembly. And so when we double click on the create average nucleotide identity comparison, the input is really just our whole genome alignment that we had just simply created in the step above. In this case, I'm not using any annotations. The reference genome I brought in was a FASTA file and really just contains ACTs and Gs. So I have no, annotations to really to sort of say, really just do the genome alignment around the CDSs or certain the gene tracks or something along those lines. So in this case, we don't have any annotations, and I'll simply open the output from this. And so we have the ANI, the average nucleotide identity, and the alignment percent. So that's the ANI up top and the alignment percent down below. And we can see here that our assembly is quite good, even without doing the polishing with the Illumina data, just because we do use the long reads to help polish themselves, where we have an average identity of 99.83 and an alignment percentage of 99.34. So these are just metrics that one would want to use to understand sort of the similarity between my reference genome and my contigs that I created during my long read de novo assembly. Now, we can definitely improve upon this. And so what we're going to look at doing now is, well, how can we do that with some of our ion torrent data? But again, just to summarize what we did to evaluate that assembly, is that we went into the whole genome alignment tool, recreated a whole genome alignment where we selected our 6 contigs from the de novo assembly. When we clicked next here, we were able to set the reference genome that we're using as our alignment. And again, I just left everything as our default settings. And that's how we were able to generate that whole genome alignment where we can visualize sort of, you know, where our contigs align to that reference genome. Right, and then lastly, we created the create average nucleotide identity comparison. And this is really what I would probably be using to look at the comparison, just because, as I mentioned, manually inspecting that alignment is, you know, pretty pointless. And so when we look at this, we had, like, you know, the average nucleotide similarity, if you will, or identity, where it was 99.8% the same. And then we only covered, though, if you look at it, slightly, you know, 99.34% of the reference genome effectively. So how can we improve this? And if we look at ways of doing improvement of these sort of genomes, there's ways of doing that when we use, you know, Illumina data. And so we've been asked, you know, to generate tools to be able to do that type of analysis. And again, if we just simply look at our toolbox here under long read support, we can easily go into our polished reads. And when you click on that polished reads, you can see it says select sequences. And at this point in time, it's going to be our contig lists. And then when we look at the polish with, this is where we're going to point to our paired data that we imported in. And you can see here that there's like a raccoon parameter, which is, you know, the size of which a partial order of alignment is performed. And so it's looking at sort of a window size here. It might be an easier way to describe it, of how it's looking through that the short reads and matching them into the contigs. It doesn't matter. And so in this case, not going to include the unpolished sequences, just because, again, I don't think it matters in this case. And I'm just going to simply save the results. In this case, we'll select the root directory, that long read tutorial folder, create a new folder called polished assembly. And now we can see it's actually taking the 300,000 reads and using our mapping algorithm, mapping them to the contigs that were created in our de novo assembly. And these reads will be used to polish and improve the de novo assembly. I guess if while this runs, by show of hands, how many people have like Oxford Nanopore data today, or have PacBio data? If you have data, yeah. And so I know I'm talking about MinION data, but these tools can be used also with PacBio data as well. Again, I'll open up the log. I always like to show people how long some of the analyses take. I mean, this isn't a huge data set, but again, this is all laptop time and, you know, live demo types of data sets. So they are, you know, downsampled to a, you know, a size that is easier to manage within the genomics workbench in these timelines. I guess by show of hand, has this been, I mean, is this at all useful? Or does it look like it could be useful for your research to have a tool to support your long reads?
Sean, so for
Mhm.
that feedback, what we can do is maybe we can just launch this poll, right? So
I
this
love
poll
it.
has like both of the questions you asked, like one was about future trainings. So to our attendees, if you feel that CLC is something that can be useful to you for your different analytical pipelines, and you want to see future trainings in other topics like RNA-seq, single-cell RNA-seq, different types of microbial pipeline, let us know through poll that is just launched. And the second question is exactly what you ask right now, Sean.
Perfect. Yeah, it'd be great if you guys could answer this, just because, we plan on doing some more of these types of trainings in the near future. As I mentioned, ATAC-seq, we have gotten asked by a couple of different organizations if we could support. And so that was something I wanted to add. And then, again, for, it's just fun to have presentations or, these types of meetings that are more useful for everybody. And so I saw there was 2 questions that had come in, and Araceli had answered them, of course, correctly as well. But yes, a lot of our algorithms are written using like SIMD, so having more CPUs and more RAM will make them considerably faster. And additionally, when I think I answered how did I know if something was a plasmid or a bacteria sequence. Not only did I show ones that were circular, but when we looked at like sort of the output, I can see like the length of the sequences. And I mean, my bacteria is much longer in this case than any of the plasmid sequence. So size would also be something that could be determinant to distinguish which one was which in this reference sequence or in the assembly. All right. Is it, we got about at 50% of this mapping, which doesn't take much longer from this step forward. And so right now we can see it's taken about 3 minutes and 3.5 minutes to sort of do some of the preparing of the data and polishing. And now it's going back through and mapping using the CLC read mapper to sort of clean up the last portion of the de novo assembly. We're almost at 100% participation in the poll. And it looks like a pretty even distribution of functionality besides RNA-seq. I'm going to simply end the poll now, as we're going to be able to jump back into the application and look at sort of some of the results that we have generated. And there was an RNA-seq training that Dev had included in the chat as well. Of course, I've already done all of this, but I was trying to do it live so that way you would have a better sense of just how long things take. And hopefully everyone's all right with bearing with me for just a few more moments to finish this analysis up.
And while we're waiting, Sean, it looks like one of our attendees had a really interesting question. They wanted to know if it's possible to assemble long and short reads in one process, or is it better to separate them like you've demonstrated here?
Well, I'm gonna get to sort of, I normally wouldn't run this as one step and then the next tool. So it depends. I'm trying to understand the question. So if it is to take like long and short reads and do them like in one assembly, I would suggest to not do that. I would use, if I had Illumina data and Ion Torrent or PacBio data, I would be using these 2 different tools. But when we talk about, and it's sort of not a bad time to do it. When we talk about importing these plugins, some of them come with template workflows. And in fact, the long read plugin does come with one. And you can see here, there's actually a workflow for doing long read and polishing with short reads. So to kind of answer your question, there is a workflow, and I would have just said, hey, do this, but then it would have taken me like, 5 minutes, and then we wouldn't have been, I just thought it would be nice to separate them, just because it allows me better time to sort of talk about each tool. And then at the end, I was going to just say, simply run this, and then you wouldn't have had to do those separate steps. But you'll still notice that there's a de novo assembly and then a polishing. So I could take that question two ways, where one way was really like, I don't even do this, polishing step, and that these short reads, if you will, right, go right into this de novo assembly, and I don't do this. Where that wouldn't, that's not as efficient, because you end up with a lot of reads in this sort of, it's just not as an efficient way of going about it within our tool. So I would simply use this workflow though, and when I run this workflow, it'll just ask me for my long uncorrected reads and my short reads, and it will output just polished contigs and a sample report. So to answer that question, I would still do it as it's still two different tools, but I would simply run the workflow.
Awesome. Thank you, Sean.
Yeah, it's all right. You kind of spoiled all my fun. That was my last slide. I was going to be like, well, why would I want to do this as separate tools? But it goes right along with that question and makes total sense to bring it up then as well. So it was just more of having fun with you, Kyle. So we can see from our polished assembly, we can look at that similar report where we can see, information about the what I really looked at, like the number of the number of, mapped reads, polished windows, and just sort of the metrics that we can see here for your sequence statistics. So again, still 6 contigs. Our maximum length increased a little bit, as well as our minimum. And so with those short reads, we were able to, clean up the assembly a little bit more. And honestly, just to do sort of a quick comparison of it, what we can easily do is come in and create another whole genome alignment, and this time incorporate our polished assemblies and our de novo assembly without the polishing with the short reads to the same reference genome. And I'll just save this real quick. All right, and so while that runs, just to kind of reiterate what we did with the, to improve the read quality, we ran the polish assembly with Illumina reads. We just selected our contigs that were created in the step ahead, and we simply pointed to our reads that we are going to be, the Illumina reads that we want to use to polish with. And we'll look at this in a moment. I guess I can just show you the output here. So if we're looking at this output, we can see our contig list up top. So that was our first run. Then we have our polished list, and then we have our reference genome. And if we look at sort of again, that ANA, so the similarity and how much of the genome is covered, what we can see is that with our polished, right, we actually improved our AP. It went from 934 to 99.57. And additionally, the sort of percent matching went up as well from 99.8 to 99.9. So we can see with those polishing, I mean, again, the de novo assembly, the first pass was actually pretty good, but we can also see that with the improvement of the Illumina data, we are able to improve that assembly. And then, that wasn't what I wanted to do. And then to the point that I had just made, when Kyle had asked, well, do we add both reads at once? And do I have to do this sort of as 2 different steps? Really, there's a template workflow that can be utilized. So that way, all you're going to be doing is defining the long reads, the short reads, and any unlocked parameters that are here, which I can show you very quickly. We don't have to wait for the results, but we can see all we have to do, double click on that workflow, And again, in the wizard up here, you can see it says select long uncorrected reads. So I know that I want to select my long min-ion reads. And now it says select short reads. So this is going to be my Illumina data. And now you'll notice that for the polished read settings, it says include polished sequences. I can either yes or no. And then I can see that there are locked settings. So the default workflow is just locked down to the default settings. And then it's just a matter of where do we want to save those results. And you could imagine that if my goal was to just simply say, hey, just run that workflow, and all you have to do is point to this and this, it doesn't really lead to a long sort of training session and give you guys a sense of all the different reports that are made and things along those lines and parameters that you have access to. So again, that workflow is just simply leveraging the tools that we had described today of the de novo assembly and the polished reads with polished reads. And then it just simply exported one set of polished content. So I wouldn't have to do this as multiple steps. One tip and trick I'll point out here is that if you have multiple tabs open, like I do right now, if you do Control-Shift-W, that closes them all. It makes it life a lot easier than closing a bunch of different tabs. All right. So one last kind of area I wanted to just talk about briefly was, how do we, maybe map our reads to a reference and visualize the assembly? And in this case, what we would want to be utilizing is the map long reads to a reference. Now I'm going to select my min-ion reads as my reads. So these again are uncorrected reads. And I'm going to simply map them to the reference genome that we brought in, which is that FASTA file. You can see that right here, reference genome. And so just some parameters here, depending on the types of organisms that you're working on, you can see that it does allow for enabling like spliced reads, if like spliced long reads. So if you were dealing with a genome that had spliced transcript, the assembler would allow for sort of gaps between where the read was being placed. That's not what we're dealing with in this case. And what we have are like the ability to change our match score, our mismatch cost, our gap open cost. So again, we try to give you guys a lot of those parameters so you can be able to change the algorithms on the change how the algorithms perform. And again, if you hover over them, it gives you sort of the range that they're allowed to be in. And additionally, if we click on our help button, we can look at how those parameters are actually sort of being utilized. So again, we can see, match score. So every match that you would get, you would end up with a plus whatever value that we put there. If you have a mismatch cost, again, whatever that value is going to be utilized as a penalty score in the overall alignment for the read to the reference genome. In this case, I'll just simply create a standalone read mapping. I'll collect all my unmapped reads, create a report, and just save those outputs. So the question about, do we need to trim or check the quality of reads? So in like that workflow, if you notice, it does have that functionality of doing a QC of the sequencing data. And so that is something that is generated and put into this sample report. I didn't talk about that today. I wasn't sure how I was going to map for timing. But again, from that de novo, or from that workflow for the de novo assembly of long reads, it would create a QC for sequencing data for your long reads. So that tool does exist in the application. I did omit it from just sort of all the individual steps that I had run. But with that workflow running, I guess we could also take a look at that QC of the data as well, if you guys would like to do so. So the read mapping is finished. So we'll take a quick look at the output that we have from the read mapping. And really, I like to look at the report first, just to get a good sense of, how many, again, we had 6 references, what was the average length, the number of bases in the as a total, how many reads mapped, right? We can see that out of the 7,000, I mapped just about 600. I mapped almost all of them, mapped 699, 6,996, sorry, which was 99.3% of my total reads, but it actually covered 99.73 of the bases. So again, it just gives me a good sense that I wasn't really missing anything in my sort of assembly, right? All of my reads are really mapping to this genome, covering this genome quite well. So it can be used as sort of a litmus test in that sense. Additionally, we can actually inspect all of the different chromosomes that were assembled. And here, again, you can see the consensus length. And so this longest one in this case is going to be the bacterial genome. And we can see that the majority of my reads are what mapped to that, which was 6,298 with an average coverage of I'll call it 15x. Additionally, we can see, other genomes or other contigs or plasmids that were sequenced here as well. If you want, we can open up any of those mappings to inspect them further. And so here, I just opened up this 638.1 mapping, and we can see here that the reads map over this origin. And just to give you a sense, when looking at sort of this read mapping, red represents a reverse read, green represents a read mapping in the forward direction. And any place that there are differences, we can see, those will be highlighted in the read. And so there are some places where we have, mismatches between the read and what is being called in the reference and in the majority of the other bases at those positions. Additionally, if we double click on the tab, we can expand that view and we can see how we want to lay out those sequences to look at, how detailed of a view. And then we can go all the way to the point where we're looking at what we refer to as a not compact view, where you can also see the quality score at any of those positions for the bases within those reads that are called. So again, just from the mapping our reads to a reference as just trying to show another tool that's within the long read functionality, we can see that all we had to do is, again, double click on our tool within the toolbox, select, in this case, the min-ion reads, and define the reference genome that we wanted to match or map them against. And we did look at the mapping parameters or options. Again, in the case that we looked at today, I didn't really have to go through and manipulate a lot of those options. And in this case, I like to create the standalone read mapping and really the report, which is most beneficial, because then we're able to, get a sense of the quality of our read mapping and our data to that reference genome. And additionally, we can explore and inspect any of those as we see fit as well. So, that really kind of got us through all of the different tools I wanted to talk about today within the long read assembly functionality. So we went and talked about just a quick overview of the application where, it was just the lay of the land that I thought would be most useful, where the import button is, where our data is stored, again, where the tools are and how we manage and install plugins. And then we went through and I showed how the long read plugin would be installed through the plugin manager and the whole genome aligner plugin. And then we talked about importing data. And in this case today, we brought in 3 different files: an ion torrent data set, a set of Illumina paired reads, and a FASTA file that we used as a reference. And then, of course, I ran independent tools for like doing the de novo assembly and looking at sort of the looking at sort of how the assembly would look with just those error-prone reads, and we did that whole genome alignment, so that way we could get a sense of the similarities and differences between what was assembled from my data and what we had as reference genomes. And then we polished the de novo assembly up using the Illumina data set. And once we were able to do that, we ran that whole genome aligner, and we were able to see that the assembly did improve, a bit. But again, we were also had a pretty good assembly to start with. But we did see an improvement in both the percent identity and, really the amount that we covered of that reference genome. And then lastly, just to talk about some other functionalities or some of the things that you might want to do, especially with having a reference genome, looking at and how to map reads to a reference. Again, everything in the application is wizard driven, should hopefully be an easy to use tool. The functions that we looked at today were based on open source tools like Minimap2, MiniASM, and Raccoon. So again, it's not like we're have a black box, but a lot of these are implemented in a way that we can make them faster or more memory efficient. And the last thing that I'll leave before I just open up for some questions for the next 10 minutes would really just be, just to keep in mind that there are a lot of applications and research areas that we can support within the Genomics Workbench. And so, if there are other areas that you guys are interested in or have projects that expand more than just long read, the Workbench could be a tool that would be useful for your research.
Thank you so much, Sean. So as Sean very nicely showed today, like in terms of long reads, scientists can do many different things using CLC Genomics Workbench. And accordingly, I'm pasting some tutorials in the chat box. So please feel free to check them out. And also another thing I'm pasting is the SurveyMonkey link. So if you like something about this training, or if you have a suggestion what we should do in future trainings, please feel free to let us know. It's a very short survey.